Hello, everyone, and thank you for joining us today for this Ask the Expert panel on debunking common myth associated with flow cytometry compensation. My name is Mirko Corselli, Global Scientific Content Manager at BD Biosciences, and I will be the moderator for today's event. It is my pleasure to welcome today's flow cytometry expert, Bob Balderas, VP of Biological Sciences and Market Development for BD Biosciences, Dr. Grace Chojnowski, Cytometry and Imaging Facility Manager at the Queensland Institute of Medical Research, and Dr. Maggie Wang, the Scientific Platforms Director at the Westmead Institute for Medical Research. Before kicking off the event, I would like to quickly go through the agenda for today's event. Our expert will start the session by providing their perspective on compensation. We'll then begin the live Q&A session by addressing some of the many questions that were submitted by you at the time of registration. I would also like to remind you that you can submit your questions at any time during the presentation live by clicking on the Ask the Question button at the bottom of the video. So with no further ado, I would like to introduce Bob Balderas that will take us through the rest of the event and introduce the rest of our speakers. Bob has 4 years of leadership experience in both academia and the biotechnology industry, and has made significant contribution as a corporate scientist, site manager, and a marketeer. He's a very well recognized international expert in the field of immunology and flow cytometry, and a key developer of new technologies in collaboration with research scientists. So Bob, it's my pleasure to pass this to you, and I hope everybody enjoys the event for today.
Thanks, Mirko, and welcome everyone. And first of all, I'd like to start today by with a welcome to everyone for having an opportunity to share some perspectives about something dear to all of our hearts. This topic is certainly something that has been debated for the 50 years that we've been in flow cytometry. Even for myself, as I look at this, I look back, 30 years, and I can remember individuals that gave me a perspective in this field. Today, though, we're challenged with many different things. The complexity of our assays has increased. The data is looked at in many different types of ways. Compensation is critical to that. We have many colors that we have to deal with, and the complexity of every single thing we do just gets bigger and bigger. I myself go back to discussions I had a long time ago with Mario Roeder, and he always shared a perspective with me when I was learning and when I was looking for answers why one data set didn't look as good as another data set. And he said, there were always rules. He had 3 good rules for even bringing in compensation controls, and I'll never forget them. He said that the controls needed to be at least as bright or brighter than any sample compensation will be applied to. And that's an important consideration to select the sample with the brightest fluorescence of an experiment. And maybe the dim ones are relatively irrelevant. He tried to share that perspective, showed me data. And he also reminded me about other things, about background fluorescence. That should be the same for the positive, and the negative control. And the third thing he always said to me was about compensation controls and fluorochromes must match the exact experimental fluorochrome. And then as we walked away, he said, there's one more thing. There's one final note, and that's compensation controls must be treated in the same manner as experimental samples. And all of those things to me still resonate when I think about it. When I look at data in the literature, and when I hand data off to have a bioinformatician really look at it, I know that compensation plays a role. And if not, lots of things can happen with that data analysis. Today, though, we have something special. We have 2 individuals that are going to share their stories with us, and we're going to address some really critical questions that always, seem to bubble up to the top. And the first speaker that I'd like to introduce is Grace. And all of us out here in Asia, and we know Grace. It's nice to have a friend, actually two friends this evening today to address this topic. And Grace began her flow career at Peter MacCallum and has been a foundation leader in flow for many, many years, and is an individual who's played a significant role in the Australasian Cytometry Society. Every year, I know when I fly into, except for last year, fly into Australia, it's always great to see her. She's known as a real domain expert in this field and a fundamental teacher in flow. And she managed, she's managed the core image facility at QIMR, since 1993. So imagine the number of scientists that she's worked with, that she brings them through a training process in her program to make sure that those fundamentals are tight and that the flow cytometry is as good as it can get. She too has been honored by the ACS for her contributions to the field. A great honor to bestow on an individual for excellence in flow domain knowledge as well as education. So it's my privilege to introduce Grace and to move forward with the first perspective in compensation.
Oh,
Grace?
wow. Thank you so much, Bob. Very kind words. And thank you for this opportunity to help debunk some of the myths associated with flow cytometry and compensation. Next slide, please. Okay, so before we do start with compensation, it's important to understand a little bit about fluorescence and, spectral emission, how a cytometer collects that spectral emission. Why do we need to perform compensation? What is compensation? And can we try and avoid having to use compensation? Next slide, please. So different fluorochromes will give you a different fluorescence emission. Some fluorochromes will only be excited by one excitation wavelength or one wavelength of a laser. Other fluorochromes will get excited by 1, 2, or 3 lasers. Conventional flow cytometers have their optics set up so we can try and collect the main peak of a fluorochrome emission. In that top graph, we can see that the FITSI emission is being collected with the 530-30 bandpass filter. And next to it, we've got the PE emission, and that's being collected by 585-40 filter. But we can also see that some of the FITSI emission is going into, the detector that we want to collect the PE signal from. And a little bit of other fluorochromes that we may have in our assay are going into that detector as well. In the two middle plots, we can see that we've got 4 fluorochromes. One, we're exciting, the one on the left, we're using the 488 laser, and only 3 of the fluorochromes are fluorescing. The Cytox blue fluorochrome doesn't get excited or doesn't absorb any energy from the 488 laser, whereas the plot in the middle on the right, the Cytox blue is being excited, but to some less effect, so the other fluorochromes that are that also do get excited by the 488 laser. In the new type of flow cytometers, spectral flow cytometers, rather than just trying to measure the peak of the band or the peak of the fluorescence emission, we collect the whole spectral emission that's coming from a particular fluorochrome. There'll be more about that later. Next slide, please. So what are we actually doing when we perform fluorescence compensation? What we want to do is subtract the emission of all of one or more fluorochromes that are actually going into a detector that from which we want to only be reading a particular fluorochrome. So if we only want to be reading the PE fluorescence in a 585 detector, we want to subtract out all the other emission that's actually overlapping or going into that detector. And it's a mathematical process where the software, if you're performing auto-comp, the software will actually subtract all the other fluorescence that's actually leaking into that detector. Next slide, please. Now, when I first started in flow, I think we were only using one colour, and then we got to do two colours. There was no such thing as compensation back then. But as the colours increased, we had to, learn to do this thing called compensation. And the principles that we used back then, whether we were doing 2 or 3 colours, is still the same with what we have to do now when we're doing, 10, 15, 20, 25 colours or more. The principle is exactly the same. Next slide, please. So what are we actually trying to do when we perform compensation? And to perform compensation, if you do have a lot of colours, take your time, don't rush it, just really try and perform good thorough compensation before you start analysing your main samples. This example here, I've got a single colour control. It's a brilliant violet 650 fluorochrome, and on the top left plot we could see that we're collecting data from that single colour where we're looking at it on a 610 versus a 670 detector plot. And what we want to do is that the fluorescence should only be seen, the positive fluorescence should only be seen in the 670 detector, because that's the detector that we want to acquire data from for that fluorochrome or from that marker that we're interested in. So if we perform compensation where we subtract a certain amount of the 670 fluorescence out of the 610 detector, the negative and the positive for the population, the brilliant violet 650, should be exactly the same. The MFI, we're trying to achieve the same MFI value for both the negative and the positive population. Now, next slide, please. So when we do this, and we've got more than just the 2 colours, we need to make sure that that 650, the brilliant violet 650, is compensated out or subtracted out of every single detector that we're going to be using in our experiment, and we should only see a positive signal occurring in the 670 detector. Next slide, please. And we have to make sure, so every single fluorescent tag that we're using or antibody that we're using, we need to perform the same thing as we've just done with that brilliant violet. So every single colour control you need to make sure that it's only, the positive signal is only occurring in that, the detector that it should be occurring in. So it's a balancing act. If you get your balance right with your 2, the weight of the MFI, it's nicely balanced. As soon as you add a little bit of voltage, then the compensation is no longer valid, and you no longer have that nice balance of with your compensation. Next slide, please. So, how can we make it nice and easy? As Bob said before, good sample preparation is critical. Good washing between after you've stained your sample is very important. titrate your antibody to work out the best concentration you can use for your experiments. Make sure you've got a single colour control for every fluorochrome used in your experiment. Again, treat your single colour compensation controls exactly the same way you're going to treat your test sample or samples. If you've got very dim markers or very low expression markers where you can't get a really nice compensation control, then use your, there's a lot of beads on the market, so you can use compensation beads if you can't get a good positive control for that fluorochrome. Make sure your instrument's clean. If there's any, residual debris or cells from the previous user, that is also going to have a negative effect on your experiment and, setting up your compensation. Next slide, please. So what we do when we set up an experiment, we're going to be performing compensation, we remove any parameters that we're not going to be detecting fluorochromes in. If you don't need them, we don't store data in them. We will run our completely stained control with all our colours, our main sample, and make sure that all the populations of interest are on scale. This is, of course, completely uncompensated, but we want to make sure that, our main population, we haven't got anything off scale and all the detectors are showing a population. We then take that off, we don't store it, we run all our single colour controls, again, make sure that the populations are on scale. We then start to acquire every single control, whether you're doing, usually we do perform auto-comp. If for some reason you've run a number of compensation controls and then you put your, your 4th, 5th, 10th one on and you realise that you need to turn the voltage down a little bit, you have to go back and reacquire data from all your previous controls. You must have exactly the same voltage settings on every single compensation control, and those voltage settings need to be exactly the same for your main sample as well. If you have performed auto-comp, usually it works fine, there's no issues, but we always check that the compensation has worked properly using an invariant plot, which you saw earlier with all our fluorochromes, to make sure everything is the way it should be. Next slide, please. So we've got quite a few instruments with exactly the same configuration in our facility, but you can't set the same voltages on 2, 3 different instruments and then expect the compensation to be exactly the same. Instruments, even if you have the same voltages set on them, are going to require different compensation settings. As soon as you've changed anything on your instrument, if it's got a new laser or you've got a new detector or you've put new filters in, your compensation is no longer going to be valid, so you will need to redo it. Next slide, please. So sometimes people will use a different marker, a really highly expressed marker like a CD8 with the same fluorochrome and try and substitute it for one that's very dim or may not be expressing. So you can get away with it with some dyes, but not all dyes. Tandem dyes, which includes the Syrigen dyes, is a big no. There's a lot of variation in the manufacture between tandem dyes, between different companies within the same company, and a lot of variations. So you're never going to be able to get the same compensation with two, just really avoid using 2 different tandem dyes for compensation. There's a really good paper in Cytometry Part B for those of you who are interested on using tandem dyes and compensation. Next slide, please. Again, don't substitute one fluorochrome for another if it's going to be, even if it is collected in the same detector. You can't use, say, something like FITC if you've got GP in your main sample. The two will give you have got different emission spectra, and you're going to need different compensation. Another one that's common that people will substitute, say, FITC when they're trying to compensate for a test sample where they're using CFSE. CFSE's got a slightly redder emissions, so you can't use FITC to compensate for a CFSE sample. You will need to use, a CFSE sample. Next slide, please. Alrighty. So here we go. If you've got a really nice, clean instrument, all your detectors are reading your signal at the right time. You're getting the right fluorescence occurring at the same time. Next slide, please. So here we've got a dirty or a blocked cell, and we've got different peaks arriving at the wrong time. We've got some of that red dye that's sticking to some of the cells that's going to give you a completely different emission on some of your control samples. And, your compensation is just not going to work. You really are going to have a lot of issues. So make sure your machine is clean. If it's an important experiment, spend a few minutes before you run all your samples to make sure, it's clean, there's no debris. If somebody's run cell cycle before you, or they've had a lot of dead cells that are stained and, clogged the machine, just take some time out to make sure it's nice and clean before you start on your experiment. Next slide, please. So how do we avoid doing compensation? Have a look at some of the spectral viewers and, have a look at fluorochrome combinations that don't have any spectral overlap. If you've got, 2 or 3 types of fluorochromes where there's no spectral emission, you're really not going to have to perform compensation. It's a little bit hard to do once you start doing 10, 15, 20 colours. You will have to do compensation, but have a look at some of the spectral viewers, and that'll actually also help you, how to pick some of the combinations of your fluorochromes. Next slide, I think that's it. Yes, thank you.
Thanks, Grace. It's always just a reminder of the complexities that we have, and the fact we're taxed with so many different types of instruments, and then when we move into what's happening in our field today with the new spectral instruments, then there's a new set of things that we have to deal with. But wouldn't life be good if we had perfect colors that didn't spill into each other and didn't have any cross laser excitation? I guess whoever can build some of those dyes are going to solve a lot of things for all of us.
Oh, there'll be somebody one day that'll invent them. Hopefully in my lifetime.
Yeah. So thank you very, very much. And we'll jump in some questions in just a couple minutes. But the second speaker today is another really, good friend. And Maggie, wherever we go and wherever we, whether it be from China down to Australia and stopping in Singapore in between, we've had an opportunity to talk about flow and talk about all the different challenges that we have. And Maggie had received her PhD in 2007 at the University of Sydney. And after a postdoc that she did at Centenary Institute, she moved to the Western Sydney Area Health Hub in 2008. And then in 2011 became a manager of the Westmead Research Hub Flow Program. But today, her responsibility has grown. And it's grown in breadth, and that breadth is that she oversees, 8 different scientific platforms at the Westmead Institute for Medical Research. And one of those one of those big platforms is Flow. And she's built a team of individuals that in her group that help in this program. Maggie too has done a lot of things similar to Grace in her career, and finds that education and commitment to a society of the ACS has just been key and centric to her too. She's served on the advisory councils in 2018 and 2019, and I'm sure they would like her to do them forever. So her career is broad. And so when we had the opportunity to try to find two individuals that shared some experience, but shared some experience in knowing each other in one perspective in the great country of Australia, but also to be able to tell her story, a different story, a different perspective. And so it's my pleasure to introduce Maggie Wang for our second presentation.
Thank you very much, Bob, for the nice introduction. Hello, everyone. I'm Maggie from the Westmead Institute in Sydney, Australia. So for those who never heard about Westmead, it is located in Western Sydney, about 25 kilometers away from city center. The Westmead Cytometry is the only flow cytometry core facility in Greater Western Sydney area, so we're all very proud of. Bob and Grace have given an excellent summary of key elements of compensation and their recommendations of the best practice. In the next 10 minutes or so, I would like to share the pain point and the best practice at Westmead Cytometry. These tips were developed over a number of years at our facility, mainly driven by the frustration from our users. Hope it would help you if you have experienced the similar issues for your compensation. Next slide, please. Firstly, I would like to share how we address autofluorescence in a multicolor panel. Cells can have autofluorescence that can result in incorrectly assigned signal in fluorescence parameters. In multicolor panel, the autofluorescence of the cells often shows a correlated distribution as a diagonal on the two-dimensional plot. Because of the diagonal distribution has been used as an indicator of a potentially incorrect compensation, it raised concerns that there may be some problem with the compensation. So this is an example from America. Samples are from human gut that have highly autofluorescent macrophages. The FMO control was gated on live HLA-DR positive, CD14 positive cells. The autofluorescent channel is against FMO channel. Sample was gated on live HLA-DR positive cells. The mononuclear phagocytes were visualized as CD14 against CD11C. As you can see, when autofluorescence was not compensated, the diagonal appearance was seen in both FMO control and the full stand samples on the left of the plot. The FISI or the brilliant blue 515 detectors was hampered. This is primarily because of the autofluorescent emission spectral is different from the fluorescence dyes. Thus, the spillover matrix used to compensate stent cells will not properly correct autofluorescence. Therefore, the autofluorescence distribution shows an undercompensated appearance. So how to solve it? We can actually remove autofluorescence by assigning it to an unused channel or empty channel and treat it as one more color. Now, after compensation, we can actually correct the contribution of the autofluorescence in all other channels. As you can see on the right of the plots, after autofluorescence compensation, the diagonal appearance is disappeared. So this process can enhance the resolution of the detection, especially for low-density antigens. This data also addressed the importance of FMO controls as gating controls, especially when autofluorescence is not compensated. So the take-home message from these slides, If your sample have high autofluorescence, it is beneficial to compensate the autofluorescence for your multicolor panel. Next slide, please. The single-stand compensation controls should be prepared and run with your samples for every single experiment. However, in some of our projects, a large number of patient samples need to be collected and prepared on the day. So preparing all compensation controls on the same day was challenging. Before deciding whether to use the same compensation controls on the different day, the validation is required because fluoroforms can change spectral profiles, especially for tandem dyes. If you are going to reuse the same compensation controls on different days, you will need to understand how long you're going to use the same compensation control for, and whether they would be suitable for this period of time, they would be stable for this period of time, whether the fixation would make them more stable, any effect on the compensation matrix, and most importantly, any impact on the resolution of the population of your interest. So for Nicole's 23 color experiment we're showing here, she fixed her base compensation controls on day 0 and reused them next day. The heat map showed the differences of the compensation matrix in 2 different days. As you can see, there are differences occurring in day 1, highlighted in red or blue. Some channels have more changes than others. Next slide, please. So knowing where the changes impact the sample is important. As you can see, there are some channels that are not impacted much, the top 2 plots, but others are. Therefore, if you plan to use your compensation control from a different time, you need to understand the effect of your population. You need to know whether you still could resolve the population of your interest. So the take-home message from these 2 slides is validation of your compensation control stability is critical if you could not prepare fresh compensation controls with your samples. The next slide, please. Okay, now I would like to share a tip from our facility. It is very simple, but very useful. It has been very popular since we introduced it because it has saved a lot of time and trouble for our users. So as you know, set up the detector sensitivity of high parameter instrument is important for compensation. It is very time consuming to manually run through 20 or more tubes and adjust voltage for every channel. So in our facility, we recommend a solution to our users to pull 10 microliter from each single stand controls and run the mixed sample to adjust voltage for all channels, including unused ones, to ensure no detector is off scale and signals on primary channels is brighter than any other channels. So both of these are important for compensation, especially using spectrum mixing method later on. We also validate the full stand control to ensure the detector sensitivity is not decreased due to overly bright compensation controls. So in this example, for Wei's 20 color panel, she used base 17 markers and cells for 3. Therefore, she had 3 samples, mixed single stand base to check whether all signals are on scale. If not, just the voltage to bring them back to scale. Then the next sample is mixed single stand cells to do the same thing as beads. Finally, she has an aliquot of the full stand samples to ensure the voltage are right for the cell population. So this method has significantly reduced compensation setup time on the instrument for our users. Hope it would help you if you haven't done this yet. So I will stop here and happy to take questions. Thanks for your attention.
Thanks, Maggie. Thank you very much.
Thank you, Maggie, and thank you, Grace and Bob, for your perspective. We can move to the live Q&A session. But before that, I would really like to thank our expert and our presenters, not only for the perspective that they shared today, but I also want to acknowledge that they spent a lot of time working with Bob and myself up until yesterday to go through the over 100 questions that we received from you at the time of registration and really identify patterns of what are the most important questions, and they tailor their presentation around that. So you might have seen some of your questions already answered. Unfortunately, we cannot address over 100 questions plus the one that are coming, but we did this work on going through it and select 3 questions from the pre-submitted list that we are going to address now. And we also want to create a little bit of an engaged and fun environment. So we're going to have a little poll for these questions. And next slide, please, Becky. So one of the common questions that we received over 10 of these questions is the usual question: what's the best compensation control? Is it cells? Is it beads? Or it really depends on your experiment. So you should see a poll coming up with this question. I will encourage you to answer the question. There's no right or wrong question answer, really. This is more just for engagement and fun. And while you address these, I will pass this question to Maggie to give her our perspective on the compensation controls.
Thanks, Mirko. This question has been also raised a number of times by our users. So at our facility for multicolor panels, we often use beads and cells for our single color compensation controls. So we found beads are often better than cells as long as the antibody binds to them properly. So I mean, match spaces or the light chains, especially when markers have low expression on cells or cells are limited from precious samples. So beads can be really useful. However, when you may not want to use beads, so for some certain fluoroforms, some example of this fluoroform including BUV 737, V500, or M-Cyan, because of the emission spectrum of these dyes is different on beads than on cells. So the spectral shift can lead to incorrect compensation. So we will need to use cells for the compensation controls for these fluoroforms. So if the marker are bright on cells and beads, for example, HLA-ADR can be bright on dendritic cells from tissues, you will need to use cells instead of beads. So other things to consider, when you use combination of the beads and cells for your single stand compensation controls, ensure not to use a universal negative control. Instead, to compare unstamped beads to stamped beads and unstamped cells to stamped cells. So beads normally requires less antibodies to stand than cells. So if the positive beads are off scale, we normally add less antibodies on the beads instead of turning down the voltage to bring it back to on scale, because it may reduce the resolution of your markers on cells. We also found some type of beads deliver more accurate result than others. That's due to potential spectral shifts. So to achieve accurate compensation metrics, no matter you use beads or cells, you will need to record enough events for all your single color control. So at our facility, we recommend our users to collect at least 5,000 events in each negative or positive gaze for high parameter experiment, I mean over 20 colors. So again, regardless using beads or cells, the validation is required. So that's what we recommend to our users at our facility. Thanks, Mirko.
Thank you, Maggie. And we can move forward with the second question. And again, you should see a poll question. Another big question, and I'm going to tie this also to some questions that came live. And Grace, this is for you.
Okay.
How often do I need to run compensation controls? Every time I run an experiment, once a week, once a month, once. And also, give me just a second, because I want to tie these to a couple of questions that came live you can address simultaneously. I don't see the questions anymore. Okay, we have a question about what validation criteria to use for day-to-day variation for compensation metrics using the same compensation stain count beats over multiple days. And maybe that can be part of the discussion on controlling the experiment over time.
Okay, so Maggie did address that a little bit just before. I guess you have to determine or ask yourself how important is the experiment that I'm running today? Is it really, really important? And if it is, run compensation controls the same day that you're running your samples. We perform quite a bit of work for external users, and we always run compensation with the test sample that we're running. Sometimes the test sample, the cell numbers are limited, so we run a external control. We get Rappicoat from the Red Cross that we use as a internal control to make sure all our fluorochromes are working, all the markers are working. If some of the markers are dim, we run beads. Sometimes we'll run beads and compensation. We'll run cells and beads to set up our compensation. If you're running the same experiment week after week, day after day after day, you will get away with, using the same voltages and the same compensation. But what happens if something goes wrong? I had a user a few weeks ago who had perfectly stained cells. He'd been running the same experiment, for weeks. And his stain sample was perfect. There was nothing wrong with his staining. There was nothing wrong with his sample preparation. But somebody had left some residual PI in the sample, and his population was all skewed. He was getting funny clusters everywhere. And I said, well, what do your compensation controls look like? And he said, well, I didn't run any. And then when he did go back and we reran everything, he would have picked up that there was something wrong with the machine before he put his main, sample on. So, how often do you run them? You have to ask yourself, the question of how important is that experiment that I'm running today? And if I don't have enough test sample, well, what else can I use to make sure the machine's working, the detectors are all working the way I want them to work for my experiment? And what was the other question, Mirko, that just arrived? Was there another one?
No, I think we addressed that. It was more about the controls over time, but I think it was addressed also by Maggie.
Yeah.
I think we can move maybe to the third question. And we receive a number of questions about this topic as well. So these are again other questions for the poll. Spectral flow cytometry eliminates the complexity of fluorescent compensation. True or false? And I would like to address this to both you, Grace, and Maggie, and give your perspective based on your experience with spectral flow cytometry.
Maggie, do you want to? I'll go. So we've got a few spectral cytometers in our facility. We still need to perform compensation after we've acquired data. Does it minimize the complexity? It does minimize the complexity, but it doesn't eliminate the need for compensation. Sometimes you still need to perform compensation after you've acquired all your data and you've performed unmixing.
Thanks, Grace. So I will, thanks. So I will share our experience because we have, for our high parameter panels, we use a high parameter cytometer, which is Symphony at the moment. So our users are using spectrum mixing method in FlowJo for compensation post-sample acquisition. So using this method, set up the compensation controls are the same as a conventional compensation. So it's not really decrease the complexity of the compensation in our hands, because we still need to prepare all single-step compensation controls. The difference is to collect information on all channels, including the unused ones, for spectrum mixing later on. However, we do find the spectrum mixing protocol give better resolutions on some channels compared to the conventional compensation using FlowJo. So that's our experience.
So with us, when we don't have the Symphony, we have the a few Auroras. One thing that is really critical when you are using spectral cytometry, you really, really need to make sure you have good, clean, well-prepared samples. And, you know, with some viability dyes, you do get non-specific, well, the negative population does take up some of the dye, or if you haven't got, you know, a good titer for your antibody, if your titer's too high, you're going to have, you know, a little bit of non-specific binding with your negative. So your negative really, well, your negative no longer is a true negative. There's a little bit of fluorescence there, and you will run into problems if your negative isn't a true negative. And, you know, even with the other instruments or lateral testers, it's a good idea not to always use universal negative. If you've got a mixed population, it's, you know, use a negative per compensation sample.
Thank you both. And I think we can start addressing, we have roughly 10 minutes left, and we would like to address some of the great questions that they're coming live. So let's try this question. This is a good one. If I'm having two sets of samples, one is control and then another one is treated set. For running compensation, are both samples needed or one control is good enough?
Yeah, sorry, say that again.
So if you have, in your experiment, you have an untreated and treated sample. Do you use both samples to compensate each sample, or one universal set of compensation would be sufficient for both samples?
So your compensation control should match your test sample. So whatever you've done to your test sample, the compensation control should match. However you've treated your sample, you treat your compensation samples for that sample the same way.
So just to confirm, you would recommend to have two sets of compensation, one compensation with single stain untreated and one with single stain treated, to apply to the different samples treated differently.
So yeah, if you're following different protocols, if you're using different buffers, if you're fixing them different ways, then you should be matching, you know, your sample with your controls.
Maggie, do you have anything to add?
I would recommend, you know, like for a lot of markers we're using beads, right? So it doesn't matter it's traded or not traded, that mainly for cells, I would say. So if the beads have a brighter expression for those markers, very used for compensation. But for the traded or untraded, I think I would scan the markers and say which one giving me the brighter kind of like result. So for the brighter one, I will use for that as a single color control. So I think just to follow the same principle for the compensation, the controls have to be brighter, or at least as bright as samples. So that would be my approach.
And actually, we have an interesting comment from Teresa. I think I just lost it. How about in this scenario, how about in this scenario mixing treated and treated sample in a single tube and run a single tube with both for compensation?
Right.
So I don't think we have done that, but look, just to thinking about it, if we mix the cells with treated or untreated, and stand the antibodies, then we would be able to catch the brighter population. And I think that would work theoretically, but we just haven't done that. I don't think I have done that myself.
No, we haven't done that.
Interesting approach.
Okay. So we have a couple of questions about beads and compensation. So I think we can go back and address some of these. For example, if during compensation, beads are lower fluorescence than cells, is this problematic?
Yeah. I mean, you've got to try and have the brightest population you can. You know, the brighter your control sample, you know, the better your compensation calculation is going to be. So, you know, if you've got a sample, if your cell sample is going to give you a better positive control with the comps, you would use that rather than beads.
For my, you know, I think for me, I would say probably it's not. It doesn't. I don't think it's a problem as long as the fluorophore behave. The spectral profile is the same on beads and cells. So basically, BUV is 737. You have to use cells instead of beads. But the background of the beads is lower than cells. I don't think it's going to be an issue. If you do have autofluorescence on your cells, and as I presented, I think it's better to do the autofluorescence compensation. That will help. But in terms of the use lower, sorry, for the lower fluorescence is actually, yeah, sorry, that means positive, right? If the positive is lower, it's definitely a problem. Probably I misunderstood it. So you have to be at least bright as your cells. So you can't use that base basically, or you may have to use a plus base instead.
Okay. And I'm seeing a couple of questions going back to autofluorescence. So maybe can we go back to the autofluorescence and how to deal with autofluorescence for compensation? I don't know, so Grace, if you have an opinion on that.
I guess, I mean, you know, when you are going to be performing autofluorescence, or you need to set a channel that's going to give you the, you know, where your autofluorescence occurs the most, and that's the channel you would assign, you know, as another detector. You know, and a lot of it depends on the cell type. Different cell types will give you different, you know, autofluorescence. You know, the flavins, the NADP, they all fluoresce depending on what, you know, state they're in the cell. You know, macrophages are a wonderful autofluorescent cells. So you just need to have a look, run an unstained sample, see, you know, where is most of the autofluorescence occurring, and then assign that detector where you're getting most of the autofluorescence occurring, and treat your your unstained autofluorescent sample as you would, you know, all your other compensation controls.
Yeah, agree. So basically empty channel, as Grace said, if that gives you the most autofluorescence in an unused channel, and you want to use that channel to assign your unstained cells for your autofluorescence as a color to add in your compensation control.
Something else that, I mean, this is sort of going to sweep a few cobwebs out from 34 years ago. We used to do a lot of runs, you know, looking at different types of chemotherapy drugs. And depending on how you treat your cells or what you've treated your cells with, some of those agents do fluoresce. I mean, if, you know, you look at your, you know, your daunorubicins, your doxorubicins, they fluoresce beautifully. And we used to do a lot of multi-drug resistant studies just by, you know, looking to see how much uptake the cells had of that drug. So, you know, something else you should be aware of when you are performing experiments, you know, what are you treating your cells with? What are the substances that, you know, you're doing your experiments with? You know, do any of them fluoresce, and are any of them going to have any impact on, you know, the populations that you're interested in?
Okay, great. We have 5 minutes left, so I would like just to take maybe a quick last question before closing remarks. And the question is about basically when you compensate on flow, your flow shows you an n/n plots for each sample. If you see that the compensation is off, is it okay just to adjust it by eyeballing rather than calculating the MFI, looking at n/n? But that leads for me to another question that always comes up. Is it okay to tweak the compensation value? When is it okay, and when you should instead reconsider maybe your control to your experiment if the compensation was not correct? So how do you deal with improper compensation?
So we always check using an n/n plot on all our instruments where all the users in the shared folder, they've got an n/n plot with every single detector on that instrument. If they're not collecting anything into that detector, then they can delete that row or that column, and you check, and you might look and you think there's something not right. By adjusting it just using just by eyeball probably isn't a good idea. Maybe if you're using 1 or 2 colors, you might be able to get away with it. But if you are just eyeballing and performing compensation manually, I don't think that's a good idea. The team in the lab, they all like to use FlowJo to, tweak any compensation that needs tweaking. I'm old school. I like to use Diva before, I go on. I like to have my n/n plots. And then, sometimes we'll draw quadrants and we'll have a look to see, what is the MFI for the negative and the positive. And then we'll go back and have a look at the population. Do we have any antibody clumping? Do we have rubbish in there? What may be causing this strange skewing, of our compensation? So, yeah, it's fine to go ahead and tweak your compensation later, but don't just eyeball it. Try and, look at it properly, look at it carefully, try and understand why the compensation doesn't appear to be right, and then try and use some maths to adjust it.
Okay, thank you so much, Grace. And we are at the top of the hour. So I would personally like to thank again our expert and the audience for the engagement pre and during the event. If you have any questions, please do not hesitate to reach out to your BD regional contact. And Bob, I don't know if you have any final remarks for closing. You're on mute, Bob.
I would also like to thank Maggie and Grace and thank those the questions that came in. I was not surprised a little bit. I think the question that came in actually about bringing samples together is something that I think we're going to see more and more of. The complexity of the instrumentations that we have today, and the fact that in spectral work we're moving. The field is moving over even 40 different colors, allow us to do different types of experiments together in a single tube. Allows us to bring a kinetic experiment together into a single tube where there's multiple events, multiple challenges to cells that are a little bit different. And we're going to have to work out those types of barcoding applications for which cells are going under different types of stress, and then bringing them all back together, in a single tube where we can probably only pick, one comp on one color. So I think it's a really good question. I think it's forward thinking, and I think barcoding is going to be something that we're going to see a lot of in the future of our work. So whether it be spectral or whether it be, the work that we do in conventional flow, pay attention to your single color controls. And at the end of the day, no matter what we do, bring a great sample into a flow experiment.
Thank you so much, Bob. And thank you again, everybody, for attending. We're looking forward to seeing you for the next session. Thank you.
Thank you, everyone.
Thank you, Mirko.
Bob. Thanks,
Bye.
Mirko. Thank you, Maggie.
Thanks, Grace. See ya. Thanks, Bob.
Thank you. Thank you, Grace, and thank you, Maggie.